Okay, so hi everyone. My name is Kaitlyn and I'm going to be presenting with Shainah. And the title of our journal today is Elevated Intraocular Pressure Induces Amyloid-β Deposition and Tauopathy in the Lateral Geniculate Nucleus in Monkey Model of Glaucoma. This was written by Yan et al. This was published in the journal titled Investigative Ophthalmology and Visual Science with an impact factor of 4.925. So here are our abbreviations that we're going to be discussing today, but we'll make sure to mention them as we go through the presentation today. So here are the contents that we'll be discussing today. We'll be going over some background, figures and discussion, conclusions, and criticisms by first going over some background. So our guiding question today is there an association between glaucoma and AD pathologies? And pathologies meaning how some sort of disease arises. So first, we're going to be comparing both AD and glaucoma. So beginning with glaucoma, glaucoma results from a significant increase in intraocular pressure, which is IOP. And this occurs because of inadequate drainage between or inadequate drainage of the aqueous humor through some sort of blockage with the trabecular network that sort of drains that. And so this can cause some sort of damage to structures within the eye, such as the optic nerve. And the optic nerve is truly a bundle of axons rooting from regions of the eye, such as the RGCs. And so this can cause stress to the optic nerve because increased pressure can cause some sort of vision deficiencies and degeneration. Now, current treatment methods have been trying to treat how can we reduce IOP, but truly how glaucoma sort of arises has been not as clear. So yes, we might be treating the symptoms, but we are not treating the cause. So here, discussing how glaucoma arises is going to be truly important in how sort of glaucoma and AD relate to each other. Now with Alzheimer's results from some sort of aggregation of abnormal proteins, such as amyloid-β and NFTs, which is neurofibrillary tangles. And so with this sort of aggregation, we may be hindering some sort of that connections and communication when it comes to visual processing. And actually some sort of, or a significant increase in doing so can cause RGC apoptosis, some sort of cell death. And now also, RGC damage is characteristic in both AD and glaucoma. So this is trying to draw a bridge between Alzheimer's and glaucoma. And also another sort of similarity between the two is that phosphorylated tau and NFT can cause damage to the LGN, which is a region of the visual or the CVS, which we're going to be discussing pretty soon. And this sort of damage can cause further reason why vision might be impaired in Alzheimer's. And so here we're going to be discussing just sort of for your own information on how the central visual system is related to today's project. So here in red, the researchers were just investigating retina, the optic nerve, the lateral geniculate nucleus, as well as the primary visual cortex. So just by noticing this figure, we can kind of see how all of these structures are related to the eye. And I'm just gonna just draw your attention to the lateral geniculate nucleus, being that the LGN, its location within the brain has close proximity to the thalamus, which is another brain or a little portion of the brain that can help relay messages throughout the entire cortex. So being that its proximity is very close, this is going to help relay visual processing or visual signals throughout the brain and to the eye. And so the researchers induced glaucoma in monkeys through the process known as laser photocoagulation. And usually this procedure is used to treat so any sort of abnormalities within the eye. And this is through like the procedure is done by first dilating a patient's eye and inducing a laser in order to target any abnormal tissue within the eye. And this helps in trying to treat any sort of like blood vessels that might be resulting in hemorrhages and so on. And so glaucoma relates to this procedure because it can cause intentional scarring. And so through this scarring, we are able to increase intraocular pressure. And actually, this is a sort of a common side effect in glaucoma. Okay, so now moving on to some figures and discussion, we're going to be beginning with some figure 1. And figure 1, basically, it verifies whether the glaucoma model is true and whether any changes are evident. And so looking at figure C, we can see that on the left side, we are seeing there is a significant decrease in the optic nerve size in comparison to the control. And now looking at figure D, we are looking at the lateral nucleus layers. And here you can see that in the glaucoma model, we see a significant like shrinkage of the cell layers, and they're not as distinct as you can see in the control. And so this is kind of giving us an idea on how our vision might be impaired through this sort of atrophy or loss of material in our eyes. And so this figure in figure E is just confirming the quantitatively on how the diameter of the optic nerve is significantly decreased from the control. So now looking at figure 2, we're going to be analyzing amyloid beta, phosphorylated tau, and in several different regions of the eye known as the LGN, V1, which is the primary visual cortex, and HPP, which is the hippocampus, via immunohistochemistry. So we're going to be focusing on the LGN specifically because the other sort of sections did not have any significance, or there was not much of an increase or decrease when it comes to these levels of amyloid beta and phosphorylated tau. But in the LGN, we can see that there is more frequent aggregation of amyloid beta in glaucoma, which is G right here, in comparison to our control, being that there's not any detection of amyloid beta. And now when it comes to phosphorylated tau, we can see that there is significantly more of this protein in comparison to our control. So here we can see that quantitatively, phosphorylated tau is significantly increased in glaucoma versus in our control. So this is just showing us that there seems to be some sort of correlation between AD and how it might arise, and how glaucoma might arise, and how it can affect our vision. And so lastly, with my figure 3, we are going to look qualitatively and how these proteins might be marked using immunostaining. So as you can see in our glaucoma model, or in glaucoma section right here, we can see that the green marker, and as well as the red, which is noon, and green being phosphorylated tau, that they're co-expressed. So they try to remain at the same level where phosphorylated tau is, noon is, where noon is, phosphorylated tau is. And so as you can see, there is way more phosphorylated tau detected in our glaucoma versus our control. And when it comes to DOPI, which is another marker, this marker actually detects retinal ganglion cell nuclei. So we would expect to have an increase or more or greater amount of DOPI detected in our control, because in our glaucoma, these RGCs are dying in our glaucoma. So all in all, reduced DOPI is just showing us that there is a loss of RDCs in our glaucoma.
So in the discussion of figures 4, 5, and 6, This portion of the article mainly talks about the presence of some of the hallmarks for Alzheimer's disease. Specifically in figure 4, they use silver staining to reveal that there are plaque and argillic structures found within this LGN section in our glaucoma model. So mainly at this figure, I wanted to focus on figures A and C. And in figure A specifically, we see that the staining provides evidence that there are neuritic plaques in this glaucoma test model versus the control. And so as we know, Alzheimer's disease is mainly found based on the presence of these plaques because of some neurodegenerative features that are found within neurons and axons as a result of the development of Alzheimer's. And so because of the fact that this is found in our test model, which is in figure A, it suggests that there is a link between these glaucoma models to Alzheimer's disease, starting with this first presence of plaque. Moving on to the next figure that we have here, figure 5 also goes into some more of the analysis of some of the different hallmark features of Alzheimer's disease that we find in our LGN models. And figure 5 specifically used a ultrastructure analysis to kind of observe how the anatomical changes within the LGN model correlated to that of Alzheimer's disease. So starting with this first section of figure A is our test and figure C is our control. In figure A, circled in these yellow, these are actually thickened myelin that you see are more abundant in figure A than in figure C. And so the significance of this thickened myelin here is basically just associated with neurodegeneration. So the increase of intraocular pressure as a result of glaucoma is actually disrupting neuronal communication between the eye and the brain. So as a result, there's also the formation of these thickened myelin and the swelling of the myelin, which prevents that communication from happening. In addition to that, we see more evidence of plaque within our LGN model, and this plaque is circled in the red. And these plaque formations are caused by axonal malnutrition, which is basically caused by hypoxia that you see in glaucoma. And so because of the fact that these axons aren't getting the required nutrition from the communication that you'd usually get in a normal non-glaucomic model, these are actually causing twists and bundles to form as you would find in Alzheimer's disease. So this is significant to its relation to our NFTs, which are our neurofibrillary tangles. And moving on to our next figure as well, figure 5B and D discuss the presence of these neurofibrillary tangles within our test model versus a control. And so figure A shows that line highlighted in yellow are the tangles being present in our LGN model as compared to the control. And the significance of these NFTs is that its presence is caused by the collection or abundance of p-tau, which is our phosphorylated tau protein. And with this p-tau, we know that these structures are forming as a result of the progression of Alzheimer's disease. And so when we take a look at the next figure, which is figure 6, we go through some quantitative testing to kind of confirm what we're viewing qualitatively in terms of our imaging that we've previously done. So figure 6 actually uses a Western and an ELISA test, but for this section, I wanted to focus mainly on the Western, essentially because they're confirming what we've already seen within the Western using the ELISA. So starting with the figure A here, we're taking a look at the presence of these proteins within 3 sections of our brain, starting with the LGN, our visual, and then our hippocampus. So as I've mentioned before, p-tau is something that we typically see with Alzheimer's disease, and its presence is going to signify that there is a progression of Alzheimer's within any model that we have. In addition to that, our A-beta is also another hallmark biomarker for Alzheimer's disease as well. So with the anatomy that we just previously saw in figure 5's imaging, we know that structures related to these markers are present. And so when we take a look at our Western blot, we can see that because the structures are there, these protein biomarkers are also there, which confirms that there is something going on between our glaucoma and Alzheimer's models. So specifically within the LGN, we see that there's actually an overabundance of p-tau, or the tau protein that we were viewing within our NFTs. And so you can see that that darker band of the tau under the LGN is much thicker and darker than what we would see in the A beta. And figure B essentially quantifies what we qualitatively see within our Western blots. And we see that although there is some viewing within our visual cortex, most of what we're viewing is occurring within the LGN, which aligns with what we're seeing with our glaucoma model. So with the summarization of these figures, we can move on to our conclusion, which essentially can be summarized within these three key findings. So firstly, with our NFT and plaque findings, we see that in our induced glaucoma model, there is an anatomical change that's occurring as a result of the glaucoma being induced, which does mimic pathologies that we do find in Alzheimer's disease. So that being said, anatomical, we can also see physiological components, which is within our A beta and p-tau biomarkers that we do find in our models, as well as some just typical features that you'd find in Alzheimer's disease. And so essentially, when you combine these two findings, the general gist of this paper is to show that as a result of physiological and anatomical hallmarks of Alzheimer's in our glaucoma models, there is something that's linking the two of them that can be further studied. And so in essentially layman's terms, we can have our take-home message in the following slide that basically answers our guiding question. So as a take-home message, we do now know that there is a link between Alzheimer's disease and glaucoma based on this pathology. And whenever we read articles, we also do want to raise questions and ask how we can better improve what we're seeing. And so this section just discusses a couple of criticisms that we have in regards to the article itself. And starting with our first one, whenever we're studying anything that requires vision or studies vision, we do believe that it's essential not just to show that there are quantitative and qualitative functions that we can see on paper, but that there is actual communication occurring. So our first criticism of this article is that the confirmation of an eye-brain communication that's occurring would actually strengthen the points that are being made. And so the reason being is that there could be a potential failure to ensure that the function is actually occurring in terms of the capacity between the communication between the eye and brain. And this is because while there may be a protein present or maybe there's an anatomical change, that doesn't always mean that there's an impairment of that specific section. So our suggestion to kind of improve this section of our criticism is essentially just to perform a functional magnetic resonance image, which is an fMRI. And by doing this, you can study the function of the vision within these test monkeys that we have and actually be able to see whether or not vision is correlating to the functions within the brain itself. As well as another criticism that we have, it's essentially just the longevity of the disease model. So the presence of the markers and the anatomy does suggest that there's a progression into Alzheimer's disease, but while this is happening within the 6-month span of the study that's occurring, what if the development doesn't actually correlate later on with the findings? And so a suggestion would be just to conduct a longer longitudinal study, possibly spanning a longer disease progression. That way you get a better exploration of the link between glaucoma and Alzheimer's disease progression. And that way you can further study whether or not this is actually going to be occurring. And so here's a couple of our references. But before we finish, I also did want to mention that in terms of the longevity, it has been mentioned that within the model presented in this article itself, it's been kind of debated whether or not there is a link. And so this article specifically was one of the first to suggest that there is such a link. And that being said, they've also found that it progresses from essentially the start of our visual processing So within the retina, all the way back into our actual visual cortex. And so again, with the longevity criticism, that's part of the reason why we also suggest this, to see if that progression follows what's being hypothesized in this article. And so thank you. That's the end of our presentation, and we're open for questions.
Beautiful. Thank you so much. It was a beautiful presentation, beautiful summary of this paper. Any questions? Questions? Okay, so well, I have questions. Take me to your first slide. Let's move on slowly. Okay, keep going. Keep going. Very nice, keep going. Okay, so I love this because this will give you and your audience a map on what you are focusing on in the study: retina, optic nerve, retinal geniculate nucleus, and the visual cortex. But the title, Investigating Central Nerve, CVS, what is that, CVS?
The central visual system.
Central visual system, and you define that, right?
Yes.
Okay.
Well, yes, go ahead.
It just that sometimes it's confusing because it's very close to central nervous system. So I would just reiterate what it means. Okay, so maybe just say the whole thing. And you can also say investigating the visual pathway, because this is really the visual pathway from the retina back to the visual center. Okay. So just to let you know, I just wanted to mention that in here they looked at monkeys, and monkeys are the most animals that are very similar to us, to humans. So this study actually, it can proceed to patients, and they can look at glaucoma patients, and they can look at their lateral geniculate nucleus, look at if there is any manifestations of Alzheimer disease. And that's something they can actually do in patients with behavioral testing. Behavioral testing meaning memory testing. They can subject these glaucoma patients to memory testing and check if they are developing any early onset Alzheimer disease. And that would be it would be awesome study actually. In monkeys and humans, the final target of visual information is the lateral geniculate nucleus. But in rodents, in mice and rats, the final acceptor or target of visual information is the superior nucleus. So it's different. But we still look at lateral geniculate nucleus in the mouse, yet it's not the final receiver of the visual information. So that's also something interesting to know. But actually this study can be very easily translated to humans. Okay, so move on. You mentioned the new N in the immunohistochemistry. So take me back to that slide, to the new N here. So what is the new N mark, or what does it stain?
I noticed that I did some research about what does noon mark, and what I found is that
yes,
it marks post-mitotic neurons. So I'm assuming it's mature neurons.
It just marks retinal ganglion cell. It's a marker of retinal ganglion cell. So what we are seeing here, this is the retina, and these circles are the retinal ganglion cells marked by the new N. Now, when we see that there is co-expression of p-tau and nu-n in the same cell, what does that mean? Or what does that indicate?
What I found is that I thought that phosphorylated tau was like totally bad, but truly phosphorylated tau is needed in some sort of amounts normally because it helps in some sort of communication between the axons, I believe.
Yeah.
And so
the accumulation is bad.
yes.
Exactly. But
Mhm.
when it's co-expressed in with nu-n, what does that mean?
I'm not totally sure.
It just means that nu-n cells, which are the retinal ganglion cells in the retina, is expressing or accumulating a lot of tau, of p-tau. Okay? So whenever you have co-expression of a marker of the cell with the target that you are looking at, it means that that target is accumulating in that specific cell. Okay? Now, you wouldn't say co-express, you would say co-localize. Okay? That's a more appropriate scientific term. So you would say p-tau co-localizes with nu-n. Okay, co-localization. This is co-localization. What I would take them like as a criticism or constructive criticism, I would say that they should have labeled other cells in the retina. We know that the retina is composed of multiple cells, not only retinal ganglion cells. So the DAPI that we are seeing, like that only like in the control, you see only the nucleus, which is only a blue circle. This is only a nucleus and it doesn't have any nu-n. What does that tell you? Is this a retinal ganglion cell? It is not. So it is not retinal ganglion cell, meaning that it can be another cell. And we don't know what is that cell is. So in the glaucoma, you see, for example, on the far left of the image, just below, you see the blue, and then there is green. Far left, go far left, here. This. So you see the DAPI nucleus, and you see green, which is tau, but you don't see nu-n. This means that this is not retinal ganglion cell. What is this cell? Is this a glia? Is this another neuron in the retina? We have no idea. So actually, not only retinal ganglion cells are expressing tau in this study, in this image. There are other cells expressing it, and we don't know what are they. So I would say they have to do a follow-up study to maybe mark for other cells, glial cells, other neuronal cells, microglia, whatever cells are in the retina, to check if other cells also express tau. Because I just hypothesize that this is a microglia. Because really microglia is the source of inflammation in the retina. And maybe microglia is expressing p-tau and like taking it to the brain. Okay, so maybe, I don't know. It's a hypothesis. So this is a hypothesis that you can take it to the lab and you can test it by staining these sections with a microglial marker, right? And check, is this hypothesis is correct? Okay, so that's here. Okay, so take me to your take-home message. Okay. So I loved what you did. You started with the leading question and then you answered the leading question. That's beautiful. The other thing in the take-home message, what you can say that glaucoma monkeys may develop Alzheimer's disease because that's what happened. They induced glaucoma in these monkeys, and these monkeys showed manifestations of Alzheimer's disease. Now, take me to your criticism. First, I would label this constructive criticism. because you are giving the criticism and you are giving the solution, the suggestion. So that's they make it constructive. Beautiful. Okay, so I loved the behavioral testing. So I loved I mean the functional magnetic resonance imaging. You can also add the behavioral testing, so they can do memory testing to monkeys to check if they actually lost memory, if they are losing memory. Okay, that will make it more or stronger. What else? Long term, yes. So the long term study, definitely, because memory loss doesn't happen really overnight, and it doesn't happen in weeks. It happens at least over months, several months. So yes, I would take it to several months at least to check if they actually develop Alzheimer disease and they actually forget. This can be easily, as I said, translated to humans. We can have like a clinical trial where, or not a clinical trial, a clinical study where we can have glaucoma patients, and these glaucoma patients, we can have several testing, behavioral testing for memory testing, and over time we can see if they are losing memory, if it's not affecting them at all. So that's a beautiful study. But yeah, that's all actually for me. So it was beautiful. Yeah.
I actually have a quick question, Dr. Hasim, or
Please.
it's kind of just like a discussion point.
Yes.
I know that you had mentioned that since this disease, sorry if you hear chickens, since you had mentioned that
Oh,
this
you
study,
are hearing dogs and birds outside. It's nice today. Yeah.
but you had mentioned that since they used monkeys as
Yes.
their model,
Yes.
and monkeys are pretty closely related to humans, I was thinking that since I mean we did mention like a longevity study, do you think that maybe like since they did say that this study is one of the first to mention this link, do you think that eventually if they were able to like further confirm it, they could just transfer it over into kind of like Alzheimer's patients or glaucoma patients and use them as that longitudinal study?
Exactly. But actually, before they do that, they have to do it in monkeys first.
Yeah. Okay. And then I also wanted
Before advancing. But see,
Yeah.
this is a little bit controversial, and I'm not very sure of the answer.
Mhm.
So, and what I think, because our clinical, our patient study wouldn't be any invasive. So it will just check their memory with simple Q&A. Like really, like you would say, okay, there are multiple exercises for memory. For example, we can show them images and they tell us like what do they remember, tell them words and tell us what do they remember like after 2 days or 3 days, 4 days. But these are non-invasive. So you actually, why not doing it? So only invasive, and only like treatments, medications that needs to happen in vitro. And that's what I'm sorry, in vivo. And that's what I know. That's my thoughts, but I'm not sure of it. I'm not sure of it.
And then I also wanted to ask, since I know that we can induce glaucoma in our like in vivo models, is there, I mean, I'm just curious, is there a way to induce like Alzheimer's? Because I know Alzheimer's develops over a span of years, and like we can quickly induce glaucoma. Is there a way to like induce Alzheimer's like fast, the same way we can do glaucoma?
So there is a mouse model for Alzheimer disease. There are models of mice, and these are genetically modified. So they either induce the accumulation of p-tau, of like beta amyloids, amyloid-β. So yes, and they sell these mice at a laboratory, Jackson Laboratory for Mice. So definitely. Now the question is, if we make another study and we link it to this study, but we ask the reverse question.
Yeah.
Can Alzheimer patients develop glaucoma? So to answer this, we cannot really we can check like Alzheimer patients, we can recruit Alzheimer patients and check their eyes for glaucoma. That would be easy, right? Because checking for glaucoma is just measuring their IOP.
Yeah.
And checking under the machine, they look at the optic disc. I don't know the name of the machine, but they look at the optic disc. So it's easy to look at this. But we can also bring that mouse model of Alzheimer disease and check their vision, their eye. So we do the opposite. Definitely. Definitely. It was a nice study. Very nice. Any other questions, guys?
I had a question, but I think you already answered it because I was gonna ask, since they said there was a link with Alzheimer's and glaucoma, I was gonna ask, do they assume if glaucoma is what causes Alzheimer's, or if it can cause Alzheimer's, or if it's like the opposite? But you already answered that for me, Dr. Hasim.
It's not a causal relationship. I don't think it's causation. It's hard to say that it's a causation. It's correlation, but it's hard to say it's a causation. Okay. Now, yes, they induced glaucoma. Control monkeys did not show any manifestations of Alzheimer's disease. Can we say causation? It's very complicated, I would say. Okay. There are multiple other factors that may affect this.
But what do you mean by correlation? Like they're just related?
Yeah, correlation means that there are other factors contributing to. So it's not only the glaucoma that induced Alzheimer's, but there are other factors. So maybe when we recruit patients, glaucoma patients, and we check for their memory to check for Alzheimer's disease, maybe some will develop Alzheimer's disease, maybe others will not. That makes a glaucoma not a cause to make Alzheimer's disease, a contributor to Alzheimer's disease. Because some humans did not develop Alzheimer's disease, others did. So some humans have the potential to develop Alzheimer's, others did not. And this play like genetics plays in this, epigenetics plays in this, multiple factors like diet, inheritance, what like if their mother actually had Alzheimer's disease. It's like glaucoma, it's also like it's inherited as well. So it is, I would say it's more complicated. So when I say correlation, meaning that glaucoma is one contributor, but not the only contributor. Okay, that way you would be more careful in this. Yeah.
So basically you're saying more like an indicator?
A
Possible
contributor.
indicator?
Yeah, you like maybe this study, what it means. Glaucoma patients may have more chance to develop Alzheimer's than like people with no glaucoma.
Okay.
They have more possibility to develop Alzheimer's. And maybe the opposite is true as well. Maybe Alzheimer's disease, Alzheimer patients have more potential to develop a glaucoma than healthy people.
Okay.
Yeah.
Yeah, I get it now.
Yeah.
But we need more like study to like
a lot of other studies and especially in humans to be able to generalize it, you know. Generalization is an issue in science, because you cannot really take one study and then generalize it to everything, right? You cannot do that.
I do want to add, just to add on to Stephanie's point, this article did talk about, I like, at least they mentioned it and they kind of discussed it a little bit, that the link for glaucoma and Alzheimer's as of right now like from what they researched before they started this was already controversial as it was, because nobody really like said that there was a link. And so because of that, like the results that they got in this study, you can kind of see it just as like a preliminary kind of thing, because there's not much else like out there that supports this as of right now. So I guess, yeah, I guess the best way to put it is just that like the results that you found within this study just kind of tells you that like they have similar presentations, meaning that they might be linked. But again, since there isn't too much backing it up as of right now, I don't think it'd be fair to really say that like they are already linked, if that makes a little bit more sense.
Yes, that makes total sense. Thank you.
Thank you so much, Shainah. Thank you so much, Kaitlyn. You've been wonderful. I have this session recorded and I'm gonna send it. And we have a YouTube channel now that we are going to add our presentations to. So yeah, thank you. Thank you, girls. Thank you. It was beautiful. Okay. Any other questions before we finish?
No questions from me.
Okay. Okay. Thank you. Thank you. I'll see you next week. Bye.
You, bye.
Guys.